Hello, we're glad you've joined us for this live webinar, Comprehensive Capture of Human Neuropathology by Multiplexed Ion Beam Imaging. I'm Alexis Krause of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by IonPath. IonPath is revolutionizing the analysis of tissue biopsies with multiplexed ion beaming imaging. To learn more about IonPath, please visit www.ionpath.com. Now, let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Sean Bendall, PhD, Assistant Professor at Stanford University of Pathology. Dr. Bendall is an expert in protein biochemistry, mass spectrometry, and stem cell biology, and single-cell analysis. His lab creates new methods for single-cell proteomics that are applied to human stem cell biology, cancer immunology, and neurodegenerative disease. Dr. Bendall will now begin his presentation.
All right, I'm going to start by thanking everybody for logging into the presentation today. A little bit more about myself. I'm a biochemist and stem cell biologist by training, and my lab develops a lot of single-cell proteomic technologies, including the multiplex ion beam imaging. And so, what I'm going to tell you today is a new area of I'm going to talk a little bit about a new area we've been investigating, where classically we've kind of looked at the hematopoietic immune system, tumor immunology. I'm going to tell you a little bit more about what we've been doing with neuropathology. And the reason why neuropathology is interesting but also challenging is because it's a really complex tissue, and it has a lot of complex issues that go along with it. And the real challenge is when you're thinking about most of our analysis, most imaging type applications you do, using fluorescence can be great, but as this slide kind of highlights here, even just doing low color fluorescence imaging on an unstained field of view, you can see you get beautiful differential autofluorescence on central nervous system tissue here. And this is because the brain is naturally full of autofluorescent compounds, and there's certain things like molecules like lipofuscin that actually build up naturally in the brain with age. And so these can be extremely challenging outside of fairly heroic sample preparation techniques to deal with. So, as a lot of people who are probably listening are aware, technologies like multiplex ion beam imaging go and they swap out the use of fluorophores for elemental mass reporters. And I tell you a little bit more about this. So originally, when working with Mike Angelo here at Stanford, we kind of came up with this idea. The short version of it is you have your tissue's mounted on a surface. In this case, we were using a analytical instrument called the NanoSIMS, and this surface here was a conductive piece of silicon. We'd mounted the tissue on it. You'd stain your tissue with your, in this case, mostly antibodies, with your metal reporter probes attached to them. We'd load the conductive surface in the instrument, and then the instrument, what it does is to generate the image at the nanometer scale, it scans the sample with a primary ion beam. This primary ion beam liberates these elemental reporters in a location-specific manner. And in the case of the NanoSIMS, it's a mass spectrometer that is based on a magnetic sector. So it had up to 7 detectors where we could measure 7 different mass channels. And putting this all together, we could then go and take those elemental reporters, reconstruct their levels and locations on the sample, and generate a histopathological image that very much recapitulated what we expected to see in the tissue. But also, in this original paper, we could actually benchmark against other quantitative IHC assays. So the good part is we could show that this was a great proof of principle for multiplex imaging. The bad part was that this type of instrumentation is extremely complicated. It's hard to access. There was we just were lucky to be at Stanford, and we happen to have one of a few of these instruments in the United States. This relatively small field of view took a long time to image, and because it was based on a magnetic sector, we only had a couple of detectors, so we could only actually, even though all of the masses, even though you could have multiple masses there, we could only collect a few channels at the same time. So this kind of brought us to thinking about new instrumentation and new applications. And what this has resulted in is Mike and I working together to prototype new types of instrumentation that is now being commercialized through IonPath, but I'm going to tell you a little bit about these early academic efforts and how it led to the new applications and ultimately to the neuropathology stuff I'm going to show you at the end here. So the new MIBI TOF instrumentation is purpose-built for doing tissue imaging, and there was a couple main innovations here. The first part on the right was kind of obvious. We went from a magnetic sector to putting a TOF-based detector. What time of flight does is time of flight lets you measure all your masses in parallel. So every time the mass spec scans, you scan the entire mass range, and so there's no limit of how many reporters we can simultaneously measure based on the detector. The other big innovation was this change on the left here around the source. We put a bigger ion gun on a 45-degree angle, and the whole point with that was to essentially sputter more material off over a shorter period of time so we could sample and thus image faster. And then we added this electrostatic analyzer in where we basically make the ions turn a corner, and the purpose for that was to essentially deal with some contaminants you often get in SIMS imaging, which are these kind of partially or ionized low energy particles that you want to kind of filter out because they generate noise in your image. And so when we put all of this together, we could then go back, and because of this new configuration, we were able to image multiple markers on a much, much bigger field of view in the same amount of time that it would take for us to get this very, very small field of view using the original NanoSIMS demonstration. So we're pretty excited about this, and on top of that, we also have an extra, layer of features we can capture, things like the SED image here, which gives you a surface analysis of the tissue that you can then overlay with your different metal-coated reporters to get these kind of pseudo-bright field fluorescence images as well. So where has this taken us? Some people might have seen this paper or maybe logged into a webinar that Mike gave a couple months back, but I think one of the really exciting early studies that this enabled us to do was instead of looking at just a couple patient samples and a couple fields of view like we did in the 2014 Nature Medicine paper, we were able to go and do a larger cohort analysis, specifically a retrospective study of triple-negative breast cancer. And bear with me. This is still leading to the neuropathology, but it kind of shows how one type of instrument application kind of leads to multiple downstream biological investigations. And in this particular study, what we did is we looked at a couple fields of view across a large number of patients, and the goal here was to really build up a rule set around the tumor immune microenvironment to see if there was anything about what was going on with tumor immunology and how it might relate to kind of disease stratification. And to make a long story short, what we found here was across a cohort of just over 40 patients, there was really only 3 flavors of tissue immune types. one tumor immune type was one basically lacking immune infiltrate, and then of the two that had immune infiltrate, they fell into these either finely mixed or compartmentalized tumor types. And the structures that we saw in the tumors were also closely linked to expression and localization of things like checkpoint molecules. And the cool part here was, after this analysis was kind of done, we went back and pulled the data from the kind of survival data from the clinical records from the patients whose samples were on this study, and we actually showed that the compartmentalized phenotype, if it was present at diagnosis, was strongly predictive of overall survival with people getting standard chemotherapy. So it was really cool in the sense that you could take something like tumor immune interaction and relate it to overall survival in a non-immunotherapy scenario. So what I wanted to give you guys a little bit of an update on is where we've kind of gone from there, because it leads into having to scan larger areas and do larger scale comparisons, which again are I think really important for neuropathology. So with this particular study, like I said, we sampled only single fields of view across a large number of patients. And one of the questions we always got was, well, what about, intratumoral heterogeneity? How does it vary across a tumor between tumor sites? And so we went back and we did a little bit of a case study here where we pulled one of the tumors from one of those original patients in the study. You can actually see here where the original tissue core was sampled from this block. And we went and we did a full slide scan on the MIBI TOF here, and what this is this is essentially a series of approximately 500 micron tiles that are stitched back together, and this scan is done at relatively a low resolution where each pixel is about 1 micron. But when we scan at that resolution, each one of those tiles only takes a few minutes, and so in a relatively short period of time, we can get a survey scan of the whole tumor and then go back and pre-select these regions here of interest and rescan them at a much, much higher resolution because, again, this technology only actually ablates a little bit of the sample each time, a few hundred nanometers of a few micron tissue section. And so once you've kind of done this, we can then go and zoom in a little bit and ask some more questions about what's going on with tumor and immune cell heterogeneity across these sites. And so just focusing on the tumor cells to start here, what we see is even within a couple of these sampled areas, you see that there's change in gradients of tumor markers like keratin, changes in proliferation, changes in abnormal expression of immune markers like class 2. And so there is a strong suggestion of heterogeneity. And when we extract the tumor cells from the image using our segmentation pipeline, we can cluster the extracted features. And within a single tumor, we can assign multiple tumor cell subtypes. And so then the question is how do these subtypes vary across regions of the tumor? And what we did is just to kind of visually summarize this. You can do dimensionality reduction, form t-SNE on this, and these are how our different tumor clusters kind of arrange in multidimensional space. And then when we reveal these tumor clusters by tumor region, what I think is this starts to highlight here is that the identity of the tumor cell seems to have a regional bias, where the regions and the tumor cells are not randomly distributed, and they seem to go they kind of cluster together. And to look at this a little more quantitatively, we can enumerate each of the cell types or each of the tumor types by region here, and you can see again there's a strong correlation between the region identity and the number of cells in it. And more importantly here, this map is hierarchically clustered, so the tumor phenotypes that are most similar to one another are close together on the map. And you can see again that there's also there seems to be a spatial similarity as well, to the point where if we actually plot the distance between tumor cells across this whole image versus the composition and the correlation between composition, there's an inverse relationship implicating that essentially the further the tumor cells are apart from one another in the tissue physically, the less similar they are. So that was interesting because, it kind of outlines a summary of tumor heterogeneity, and it actually shows that you get a phenotypic kind of evolution across the tumor that is much in line with how we think you get also clonal evolution within a tumor. Now, what about the immune system? So we went back to those same fields, extracted the immune cells using the pipeline we laid out in that original paper, clustered them using flow sum to get our different immune cell types. And just to kind of, make a long story short here, we, again, we get very nice separation of our different immune cell types. But unlike the tumor cell types in this case, what we saw was that the immune cells, with the exception of one region that seemed to have a lot of neutrophilic infiltrate, the immune cells were evenly distributed across all the regions, and the phenotypes were evenly distributed across the regions. And so this raises kind of an interesting point that even though the composition of the tumor was phenotypically variable, how the immune system was coming in and interacting with the tumor at all those different points was quite conserved. And so I think there's, I think it bodes well for undersampling tumors for studying tumor immune interaction, but it also reinforces the fact that if you want to study tumor heterogeneity itself, you have to sample much larger areas. So, like I said, we were actually coming to talk about neuropathology today, but I kind of wanted to give you an idea of what kind of a more complete story looks at when and something that's leveraging scanning and rescanning large fields of view. So what we're doing with the brain is, my lab is particularly interested in Alzheimer's disease, but also collaboratively through the chair of pathology here at Stanford, Tom Montine, we're also interested in doing a lot of Parkinson's disease research. These two diseases in particular are associated with mainly the midbrain and the hippocampus for Parkinson's and Alzheimer's respectively, and so this is some of the example data I'm going to show you today. A lot of what I'm going to show is proof of principle, and I'm going to show you a little bit about the data and the optimization. But to just kind of more broadly speak about what we're trying to do with Alzheimer's in particular, one really interesting study that we're working with folks at the National Institute of Aging on is getting at this idea of cognitive resilience. And so one of the things that, coming in as someone who has more of an immunology, stem cell biology background, one of the things I was really surprised to hear is that, neuropathology and that's associated with Alzheimer's disease doesn't necessarily always lead to dementia. And so, working with some of these ADRC, Alzheimer's Disease Research Consortium samples, what we've identified is that there are within these patient populations, there's people who, at the time of death, when they those that have donated parts of their body to science, you have their postmortem neuropathology done, and if they have no beyond regular aging, if they have no neuropathology present for Alzheimer's disease in the hippocampus, this person almost certainly never has dementia. However, there's often cases when you do have neuropathology where it is typically associated with cognitive decline, and so this person will have documented cognitive defects at the time of death. But there's also a subset of patients who present with significant neuropathology who actually, at the time of death, were not cognitively impaired versus what would be the expectations at that age. And so it really provides a clinical backdrop to start to untangle kind of symptomatic neuropathology from mechanistic neuropathology. So what does the brain look like that's, in terms of how do things change that are just associated with the process of Alzheimer's disease versus what are the changes that are required for Alzheimer's disease to manifest in terms of cognitive defects? Or possibly, what are the changes that could manifest that might be compensatory or corrective to maybe avoiding dementia? And so we're hoping that by comparing between these different patient groups, we can start to answer these things, and at the same time, by knowing their underlying genotypic status, we can see if there's anything in these processes that are related to, say, having the risk gene ApoE4. So how we kind of put this together? This has been a bit of an odyssey in the lab. We have a really great team of people, of staff scientists, graduate students, and postdocs working on this. And the goal here, and you don't need to really memorize these panels here, but we've spent a lot of time trying to screen and validate FFPE qualified neuropathology antibodies for use in multiplexed assays. And we've put together two panels to really help us kind of target Alzheimer's disease versus Parkinson's disease. And these two panels aren't unique. There's about just over 50 unique things kind of here between the two panels. There's quite a bit of overlap. But to kind of get at the broad themes of some of the things we're trying to capture here, obviously we're trying to capture neuronal diversity. We're trying to capture things like synaptic markers, both to get an idea of synaptic flavors, but also just an overall idea of synaptic density, because loss of synaptic density is often associated with cognitive decline. Multiple markers of vasculature, so that we can get an idea of those types of structures in the brain, where some disease-associated features might be close or far from vasculature. Vascular defects, again, have been potentially associated with some of these disease processes. Obviously, starting to look at the other, well, I don't want to say accessory cells, because I'm actually quite excited about the immune cell and glial cell populations in the brain, but capturing your populations of astrocytes, oligodendrocytes, and importantly, microglia. And the things that are really unique in these two panels is going after is making sure we can image the disease-associated proteins with things like AD plaques or Lewy bodies, as well as looking at the protein products of some of the risk genes like ApoE4. And so, I think one of the things that was challenging early on with this is validation of these markers. With a lot of the tumor immune stuff that I talked about earlier here, we were able to use a lot of control immune tissues like tonsil and lymph node to kind of go and use the known geography, known shapes of cells to kind of validate things. And it's not that you can't do that here, it's just a lot of the structures in the brain, particularly some of these things that were non-cellular features, things like the vasculature that I'm showing you right here, don't have typical shapes. And so it took a little bit of extra elbow grease to kind of get these things going. So the strategy that we took, for better or for worse, is again, trying to leverage pre-qualified antibodies that, people have already established and used widely, get the metal conjugation on them, and then what we would do is we would do serial analysis or serial sections of the same control blocks, where we would take one section and we would do regular single color DAB staining that you can see here in brown. And then on the serial section, we would then stain with the metal conjugated antibody alone and run that by multiplex ion beam imaging to then look and see if we got the same map, got the relatively same distribution of the marker in the same shapes with the same signal noise, and we'd kind of titrate things accordingly. Other markers, again, disease-associated proteins, which can be a little challenging to see, or plaque-associated proteins. Plaque-associated proteins are always challenging because you don't always know what the expected plaque density is going to be. And so again, making sure that you have serial sections with both the AB staining and standard colorimetric staining so you can enumerate those things. And then when we look at the multiplex ion beam imaging data, making sure that these things kind of coincide with one another. The other nice thing you can do with multiplex imaging is it's pretty guilt-free or worry-free to throw in an extra marker that you might have already validated, something like IBA1 here, which marks our microglia, and we can show that a lot of the ApoE staining, for instance, is showing up in those microglia as we might expect. The last bit here is again kind of going at some of the neuronal markers, and the neuronal markers are interesting because even within the body of a single neuron, you can get differential distribution of these proteins like vGlut1 and vGlut2. So vGlut1 is expected to kind of stain almost the entire neuronal body, where vGlut2 is a little more, I call it perinuclear, but it's the soma. It's kind of the space immediately around the nucleus. And again, to really kind of know that you are show that you know what's going on here and that things are working properly, you need to be able to kind of stain these things and scan them at the same time, but also scan them at a reasonable resolution so you can see if something's perinuclear versus more diffuse in its staining pattern here. So what does this look like when we put it through the whole workflow? And here what we're showing you are actual FFPE midbrain samples that have been sectioned onto our conductive slides. And what we're showing here is in the top is the pre-rastered one, and then again, like I said, each scan we take only shaves off a few hundred nanometers of tissue, and these are 5-micron sections, and so you can see after the fact the sample gets a little discolored, but there's still a lot of material left on the slide. And so we zoom in and we can kind of see where these two scans are, and just to kind of orientate you with what we're going to be looking at, what we can do is kind of look at these scans in terms of the restitched images. So much like we did for the tumor sample previously, the way this works is we image a whole bunch of 500 micron tiles, and so we queue these tiles up such that the 500 micron tiles are roughly 500 pixels by 500 pixels, so you're dealing with a resolution of about 1 micron. And these here, as it says, are is an array of 252 tiles, and we're imaging each one of these tiles in about 4 minutes. So it takes a little while, but not too long. It's something you can queue a few of these up overnight, and then you have your survey scan, so you can go then and rescan your areas of interest. And so the images on the right here, what we're showing you is just one channel, where all the white dots in the tissue region there are DNA-positive nuclei. And that's just to kind of orient. So what happens when we start overlaying our markers? Well, there's kind of multiple levels to organization in the brain in particular that I think is even more, much more interesting than tumor immunology even here. So we're going to start here with kind of this macro view, and there's some things to pay attention here with this macro view. And when you're judging kind of overall neurophenotypic status, looking over a large area, understanding the patterns and restriction of marker expression you get is a nice thing for validation, showing, comparing region A versus region B. But also, again, like I said, for neuropathology, looking at things like gain or loss of certain neuronal subspecies, synaptic densities, vasculature. It's good to be able to do this in a much larger area rather than just sampling, a couple millimeters worth of image at a time. So in this case, this is just showing the midbrain. The slice of the midbrain is really the panel on the right here. And in particular, what you can see here is myelobasic protein marking a lot of the myelinated material, and then TH here. This is one of those ones you'd want to see on the macro scale for Parkinson's disease. TH is tyrosine hydroxylase that marks our dopaminergic neurons, and you can see we can get an overall kind of view of density of those in the midbrain region there. And then GFAP's an astrocytic marker, and that's contrasted by VGAT, which is a general kind of synaptic vesicle marker that again is giving you an idea of synaptic density. You might have noticed, and this is something that I thought was kind of pretty, but it's kind of like, what the heck is this? This funny pattern you see here highlighted in the white arrow. What this actually is this is the optical nerve orthogonally coming into the midbrain, and you can see it breaks up the tissue structure there a little bit. So we can kind of go to level 2, which is I call the micro level, and the micro level is where we start to see things a little more one cell at a time. Again, you can look at TH, and we can again get an idea about more individual dopaminergic neurons. We can start to look at like the relationships between things like GFAP on astrocytic bodies and CD31 marking the different microvessels, as well as starting to look a little bit more closely at the distribution of microglia marked by IB1 and CD45. The other cool thing, and I'm going to come back to it at the very end, is we can start to look at things like pre and post-synaptic markers like synaptophysin, PSD95. So synaptophysin marks most presynaptic bodies. But and PSD95 tends to, even though these are archival samples, they tend to be positive on, as I understand it, on synapses that have recently fired, and you get the postsynaptic region being positive for it. So it's kind of cool here because we can kind of go down to the nanoscale here, where we take one of these tiles and zoom in a little bit more on it, where now we're dealing with pixels that are more on the order of 200 to 250 nanometer resolution, and you can start to see the coincidence of these pre and postsynaptic markers, where either they're overlapping or often just offset a little bit from one another. Going over to the hippocampus, we can kind of follow the same progression. So again, on the hippocampus, you can see the kind of characteristic shape here. It's a lot easier for me to identify, though it's still tricky. Sometimes I need to be told by the neuropathologists we work with which way I'm looking at it at. But one of the hallmarks here you can see, and again, this was acquired much the same as we did the midbrain region here. One of the interesting kind of macro views is looking at something like the disease version of the tau protein. And one thing you can see here is with the by kind of quantitatively showing the staining in the image, you can see this nice tau gradient where it's high in the CA1 region and reduces. You kind of go around the corner towards the dentate gyrus here. You see this gradient of the disease form of tau, and that is something that normally, as I understand it, accumulates with aging, and this is something that gets accelerated when you have something like Alzheimer's disease. And again, on the left and right here, you can see the just kind of distribution of the different neuronal markers, and you get things like VGLUT2, which is the perinuclear marker that marks the neuronal band going around the hippocampus here as well. And again, here on the left is just showing you a light-based image of the area that was scanned that's been mounted on our after staining on the gold slide. So you can have also a visual picture of the tissue that kind of goes along with these multiplex imaging files as well. So what happens when we zoom in a little bit? So we can go in, we can kind of pick regions of interest, and we can rescan. The area here that we're scanning now is closer to this is closer to half micron resolution, where we're looking at 500 micron tiles, and we're scanning at approximately 1,000 pixels by 1,000 pixels on a 500 micron tile. And here again, you start to reveal features of more at the more cellular level. You have markers marking. You have things marking neurons like NEFH and CD56. In the middle here, this is interesting because we can start to see things like individual tau tangles showing up and their relationship to Alzheimer's-associated plaques marked by A-beta and A-beta 40. And the other kind of interesting thing is you start to look a little more closely, and you can see things like glial cell markers like astrocytes and how they're localized close to vessels indicated here by endoglin CD105. And to zoom in a little bit more, we can kind of focus on one tile where now we're acquiring. This is scanning at our highest resolution setting, which is closer to about 250 nanometer pixels, and you can really start to reveal the granularity of what's going on. And this is looking right in the kind of the dentate gyrus area of the hippocampus here. And just a few interesting little bits to point out. We've got transcription factor, again, marking the neuronal nuclei, and you can start to see kind of the distribution of microglial markers versus a lot of the neuronal processes in that tissue. But I think my favorite picture is when this is just looking at a couple colors at a time, but when we look at a few other channels, you can see, again, the neurons there marked by the purple VGLUT2. One of the interesting things here is you see this, all the holes in the tissue. Again, this is vasculature and microvasculature, and you get a really nice idea about how all of the astrocytes, wrap around this vasculature to help form the blood-brain barrier. And this is something that we're playing around with the idea of how we can kind of capture and quantify these things and understand how intact features like this in the tissue are and how they change with our different patient control groups. One of the other things I wanted to talk about quickly before before we kind of end off is what are some of the things we're thinking about doing analytically with this data. So to kind of contrast with what we showed originally with the tumor immunology stuff, the tumor immunology stuff and the tissue immune work is relatively more straightforward in that I think there's no disagreement that, identifying and segmenting cells and extracting those cells, understanding their identity, and then relating that identity and other expression features back to spatial information in the tissue is a really nice workflow. With the brain, as you guys saw from a lot of these images, while you can see nuclei and we can see individual cells, a lot of the brain is not necessarily extracellular tissue, but it's only parts of cells, because not all the cells, some of the cells are quite large and they exist across multiple planes in the brain. And so it's, instead of just extracting cells, which I think is important, we also are starting to think about what other features do we extract. For instance, like the vascular and blood-brain features, or in this case, something like synaptic features. Because while assessing overall synaptic density is important, it's also probably interesting to just extract synapses the same way we would extract cells and treat them like populations, understand what the composition localization is, because that might have some information in there too. And then the good news is for this, just like it was with a lot of the antibodies and reagents that we're leveraging to do these assays, is that people have thought about this problem already, and they've thought about it using fluorescence technology. So this is just one example of a paper. This is something Brian Cannon, a PhD student in my lab, is working on, where we're taking these computational pipelines that have been built out for light-based imaging and enumeration of things like synapses and trying to apply them to the MIBI data. And again, to make a long story short, what this does is it essentially goes through and leverages the fact that we can image these things simultaneously, and that you should see either direct pixel coincidence or pixel neighborhood coincidence of these features in an image. And so then what we can do is we can go in and define our synaptic markers, for instance, like synaptophysin and PSD95 here. We can go through and the algorithm can assign on a kind of pixel by pixel basis a probability of whether or not it's a synapse. We can then, using our quantitative field and the qualitative field and the quantitative score that we get, we can then go define cutoffs and have things actually assigned as being part of a synaptic event or not. And so in this case, this is just zooming in and showing you features where we see synaptophysin and PSD95 co-expressed, and the white pixels have been kind of enumerated here as being positive for a synapse. And so again, this is just an idea where, just like you would segment cells from an image, we can segment and identify other interesting neuronal features like synaptic bodies or vesicles that we can also kind of extract and compare between our patient groups to see what the differences are in composition, localization, and abundance. So with that said, this has been a lot of work by a lot of people in the lab. We've been developing the instrumentation here for quite a while, and all of the MIBI stuff is done in collaboration with my partner in crime, Mike Angelo, who's right down from the hall from us. We work with Stephen Thompson, who's a very charming physicist who helped us design a lot of this early instrumentation and kind of make it a reality. Liat Karen is a talented computational biologist in the lab who's done a lot of the early analytical work on the tumor immune microenvironment stuff, and a lot of the work with the brain data. We've been leveraging her pipelines to also do analysis there. Kazalia, as you can see, with her holding the picture of the brain there. She's the captain of the brain team. This is unfortunately an outdated slide, so I don't have pictures of some of the other brain team folks, but we've got JP Olivieri, a postdoc from McMaster University in Canada, Dunya Mirden, who came to us from Europe, and she's here working between my lab and Tom Montine's lab, and Brian Cannon, who's the PhD student in our computational systems immunology program, who's been working on the neuronal stuff, and Marc Boisset, of course, who helps keep everything MIBI running in the lab as well. And with that, I think we have time for some questions.
Thank you, Sean, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question, and click on the Send button. Sean will answer as many questions as time permits. So let's get started. Our first question is, can you provide more detail on the sample preparation, such as section thickness?
Sure. Yeah. I mean, that's an interesting one with the brain samples in general. So everything that we showed here in this presentation was 5-micron tissue sections, and that, as I understand it, working with the neuropathology team at Stanford, is pretty standard. One thing we've been playing around with more, but I don't have any data to show for yet, is playing around with thicker sections where we're trying to kind of capture cells that don't necessarily sit in plane. I think you probably noticed in some of the images with a lot of things like microglial markers, you only saw bits of cells coming through, and that's because we're only imaging a few 100 nanometers at a time. So I think with potentially with thicker sections, you could image more and get the whole cell body, or even do some stuff that I didn't show here today, but we're even playing around with things like 3D reconstructions. But for now, pretty standard IHC protocols seem to work well with this.
Now, Sean, our next question. What are the biggest challenges with data analysis?
I think with the data analysis, particularly with the neuropathology, I was kind of alluding to it at the end of the presentation there. So like I showed at the beginning, with something like the tumor immune microenvironment and epithelial tissues and immune cells, when you image a plane of a sample, a two-dimensional plane, you can pretty readily, in those types of tissues, identify cell nuclei and then extract cell, an entire segment of a cell based on the nuclei and cell borders, and that allows you to extract most of what you've imaged in one of those sections. And so then once you've extracted a single cell, you can then kind of feed those single cells into, I don't want to say run-of-the-mill, but already established kind of single-cell analysis pipelines where you can then add your spatial information as another dimension into the analysis. With the brain material, I think one of the things you might have noted when looking at the kind of survey scans of the hippocampus and the midbrain, when we looked at the nuclear markers, the data is, in terms of cell nuclei in the brain, they're quite sparse. There's a lot of non-nuclear, non-perinuclear material there, right? So segmenting an intact cell out of the brain, you can do it, but when you do that, there's a lot of material left unsegmented. And so I think one of the things we've been thinking about is what are those other things? What are those other kind of microscale, nanoscale features that could be related to neurobiology, neuropathology, that don't fit into a traditional cell segmentation box? And so that's where we're starting to think about leveraging multiplex features to extract things like synapses, extract things like disease-associated proteins, so going after like Lewy bodies and plaques, and both annotating them as being there or not there, but also starting to quantitatively look at their composition with respect to the relevant proteins that form them. Excuse me. Also, using markers like CD31 and endoglin to get at the vasculature and the types of vasculature, and then starting to ask questions around what's the localization of some of these other features like cells or like disease-associated proteins to the vasculature. And so it's definitely, I think my intuition is that the neuropathology is going to be more challenging than the standard tumor immunology stuff, but I also think it's kind of exciting because, working with the neuropathologists that we do, there's a lot of stuff outside of just looking at neurons that they're excited to look at and see if it's associated with different disease groups here. And so I think, employing, these same segmentation pipelines, but thinking about new things to segment is going to be one thing. The other thing that's really wide open is going, pure data-driven analysis, taking machine learning approaches and using sort of computer vision to find other features we might not think to segment out of the image to start comparing our disease groups as well.
Now our next question, can you look at other metals that could be involved in neurological disease?
Yeah, that it's funny. I get that question a lot when I've been showing this data, and the short answer is yes. I don't think you can look at. I'm not sure if we can look at every metal because different metals have different ionization potentials. But one I've gotten asked about a lot is iron, And I didn't I didn't have the data in this presentation, but in that same paper we have in revision right now, one of the things that we showed is when you're looking at various tissues, you can see high levels of iron in tissue-resident macrophages that are I think essentially consuming a lot of red blood cells, and then we see a lot of protein components that would be associated with those same macrophages and red blood cells in those same spaces. And so I think the short answer is yes. I think the challenge with looking at other endogenous metals, especially metals that are expected to be in biologic tissues, but also more higher abundant in the general environment, is just being really careful with sample preparation and stuff like that. Because one of the things we were worried about is, we use steel blades to cut our tissue sections, which have iron in them. And so we want to make sure that the iron we're imaging is iron in our sample and not iron that we've introduced through some means. But yeah, I think that's really, I think that's a really good question. I think that's something we want to look at. And the good news is, with the imaging instrumentation, we're basically collecting the entire elemental mass spectra from, 1 to 200. And so we can pick any mass channel that we want to extract for a certain isotope and see if that isotope's there in a reasonable abundance.
Now, how many signals at a time can we measure from the slide?
Yeah. So in terms of in-house testing that we've done, I think we've gotten about 53 individual isotopes working. I don't know the exact number for what's commercially available, but I think it's somewhere around 42 or 43 mass channels can be kind of purchased and used. All the data I showed you today was, I think, approximately 40-plex stain. And it's kind of one of those things where there's a diminishing returns with some potential new metal channels you can use, because basically, there's a lot of things we can measure where we don't necessarily have the chemistry to attach them to a probe in the right form. Like we could use, we can measure iron, like I was just saying, but we don't have a good way to use iron as a reporter because I can't attach iron to an antibody very well. But in terms of things that we have good chemistry for that work with commercial reagents, most of our experiments are in the 40 to 45 plex range.
Now, Sean, our next question actually has a couple questions, so we're going to start with the first one. How long does it take for imaging the 252 tiles?
That's really good. That's a good, yeah. So I really tried my best to kind of state imaging times as I was going through. But yeah, there is an inverse exponential relationship between imaging time and imaging resolution. So with those 252 tiles where we're imaging at like micron resolution, each one of those tiles takes about, on our setup with our kind of exposure time, takes about 4 minutes a tile. So I should have this number written down, but you're looking at a couple of hours per survey scan. And so, like I said, we usually queue up some of those tile runs overnight. So about 1,000 minutes, however many hours that would be for a 250 tile scan.
Now, the next part of this question: what is the maximum multiplexing done in neuropath studies?
I mean, I think you just saw it. That's like I said before, we've got chemistry working for about 53 simultaneous reporters that we can probe for in the lab. For the most part, though, the majority of experiments sit between 40-plex and 45-plex, where we've got one isotope assigned to one antigen. I think there are ways to use spatial multiplexing and barcoding to push that higher, but if you're assigning one antigen to one reporter, where most of our experiments sit around the 40-plex image analysis range.
And the last part is, you mentioned imaging plaques was challenging. Is there any picture that you can share of plaques with MIBI?
Of a plaque itself?
Yeah.
Yeah, there was, I mean, again, in that presentation, there was a few plaques in there. I don't think I had any zoomed in pictures. There's some stuff. So all the data that I showed today was really optimization data for a paper that we're putting together right now, where we're looking at control healthy and established disease tissues, just kind of as a proof of principle for seeing if we can reveal either expected or potentially new disease hallmarks when looking at things like AD and PD. I don't at this moment have a ready zoomed in photo, but I think if you go back in or contact me directly, I'd be happy to, see if I can dig something up to show you more, or even send you a preprint of the paper where we are looking at higher res versions of this stuff.
Now, Sean, how long does a scan take?
So I think that was kind of asked already, but in terms of the scan, there's kind of multiple levels of how long does it take. So for the survey scans at micron resolution, we can scan at 1 to 2,000 pixels per second, where a given tile only has a few thousand pixels in it, and so those can be scanned relatively quickly, right? For the higher resolution scans that we were showing near the end, where we're looking at the DNA gyrus, scanning at about 250 nanometer resolution, because the ion dose is much smaller and we're sputtering less material off the slide, we tend to dwell longer with each pixel. And on top of that, we're making more pixels because of the higher resolution image. So in those cases of those 250 nanometer images, we're looking at, I'm literally typing this out on my calculator right now. We're looking at somewhere around about an hour and a half per tile, because we're dwelling for more like 4 to 6 milliseconds per pixel. So it does take longer to do those ones, but that's why we kind of like this kind of scan-rescan approach, because with the brain just looking at the gross neuropathology, it's sometimes difficult just from the slide to kind of pick the right area to scan at high resolution. So we'll do that survey scan in 40 dimensions, look at relative areas of interest, and then go back and we can rescan kind of the next layer to make sure that we're hitting the right anatomical features. Yeah, so it can, like I said, we can generate a survey scan of one of those tiles in minutes, or we can scan it at higher resolution, at higher sensitivity, and do it a little bit slower, but you generate kind of richer images, like I showed you there at the end.
Now, Sean, we have another 3-part question. The first part is, do you only use markers for different cell types?
I would say, well, no, we don't. I think a lot of the markers we use didn't necessarily go after just different cell types. I think the way we typically design these studies up front, and the way we went at this one, is we tried to think thematically about what aspects of biology are involved in neuropathology and neurodegeneration, right? And so this is where we thought there was like an immune or glial cell component, so of course we got markers for those cells. There's obviously a neuronal component, so you have to mark all your neurons. But then there's all of these things where you've got, things like synapses, which are parts of cells, but are almost an independent regulatory feature. So we start, adding in synaptic markers. And then you start to get into kind of non-synaptic things where we start, like the previous question mentioned, we start looking at proteins that are associated with plaques, as well as proteins associated with vasculature and so on. So it's not just about capturing cells themselves, but for whatever your tissue is and the experiment is, you want to capture other facets of biology at the same time, because I think it's not just about enumerating and quantifying all these things independently, but in the spatial context, understanding how they might be related to one another at the same time. And so you got to kind of do that. The other thing that we've done and driven for a lot of these analyses is we kind of pair up with a lot of research studies, which I didn't kind of go into today, where people are using orthogonal technologies, things like nuke-seq and single-cell RNA-seq. They identify new genes that might be associated with new cell types, and then we try to devise probes against those new genes or new cell types to again put them back into the context of the tissue we're already imaging.
Now, how do you use histological imaging to do confirmation of cell types?
Yeah, so I mean, most of the time it's pretty straightforward. We usually go back to the original FFP blocks that we got our cell types off, our images off of, and we'll take a serial section, and then we'll often probe a serial section of that same block and either use the same reagent with chromogenic stains, or we'll use a chromogenic stain itself, like silver staining, or something that you'd more traditionally use to kind of identify cell types by morphology instead of a marker. And so we kind of try to use these orthogonal complementary techniques but on the same sample substrate.
And then the last question is, how do you do image overlay?
So for image overlay, the good news is, so for the serial sections, it's kind of hard to do. You know, for if you're talking about it in terms of some of the controls that I was showing there, you know, we can't really take one section and image it by MIBI and another section do DAB staining and expect to overlay them and match things up. For all the other images I showed you, I think the important thing to note, just in case there was a misunderstanding here, is there is no image overlay. All the features and all the parameters are acquired simultaneously in the same pixel. So the image off the instrument is a single image where the different layers of the images, the different layers of the image are just the different mass channels we've extracted, and there's no overlay or scooching of multiple layers involved. The only kind of overlaying and alignment that we have to do, if you were getting at for this, is for the tiling. And for the tiling approach, this is something where, to full disclosure, I'd say the jury is still out on it. We are playing around with two types of tiling. One type of tiling, I'll call it clean edge tiling, where we literally tile edge to edge and we don't have any overlap, and then we count on the accuracy of the stage and the instrument stepping to be good enough that we can basically put the squares back together without any overlap. That's looking really promising, but you still get some artifacts where the stage doesn't step perfectly and maybe things are a little bit out of line. The other type of overlap we do is how most of the images were generated here, which is we specifically build in an overlap of a few microns on kind of the right and lower edge of each tile, and then we have a little algorithm that kind of overlaps and aligns those things back together based on high abundant markers so we can stitch those multi-tile images. And then we use some pretty standard noise removal approaches to kind of take the extra signal out of the overlap part, which is, again, this is nothing that we invented. This is really borrowing from the toolbox that people who do regular light-based microscopy have been using for years.
Is the conjugation chemistry you are referring to based on the ion path kits?
So so for the images that I showed today, all the reagents that we used to generate those images were all used. The reagents that we used to generate the images were all created using the ion path conjugation kit. Yeah.
Now, for the images that you showed, did you apply the denoising, the background subtraction algorithm that was published in the recent Cell paper?
Another really good question. So actually, a lot of the images I showed for the brain data had very little denoising done to them because the noise filtering in the Cell paper was a k-nearest neighbor. So that works really well when you've got a lot of kind of signal concentrated to like a cell body or a nucleus or a membrane. One of the issues, so not all, so some of the images were denoised using that with some of the more abundant, widely expressed markers, but then some of the markers like the synaptic markers that I showed near the end there, those weren't denoised at all because those are actually more sparse markers where a lot of what we think is real signal might actually get removed during denoising. And so instead, what we're using for those images is more a feature extraction-based approach, where we look at coincidence of multiple markers that we expect to be within the same pixel or within the same pixel neighborhood, and we kind of use that to identify and extract the features. So it's a little bit of both. But I think the good news is with a lot of the newer instrumentation we've been working with, and the optimization of the assays on the front end, there's really very little noise in the raw data that we collect. And so again, if you went back and looked at those neuronal synapse images at the very end of the presentation, most of the pixels are black because there's just no signal there. But a good question.
We have had some amazing questions today, but it looks like we only have time for one more. So we'll finish with this one. Do you use any matrix for the TOF? And if so, is there any matrix-related issues on FFPE tissues for non-specific signals?
Yeah, so for the matrix thing, the only matrix in our assay is the tissue itself. I think the person asking the question might be making analogous comparisons to something like MALDI imaging. So MALDI imaging is a soft ionization approach where you're looking at kind of de novo proteins in the sample. And for that, you do overlay a chemical matrix on the sample to help preserve larger molecules during the ionization analysis process. In our case, all we do is a standard IHC workflow where we've got our element conjugated antibodies, they stay in the tissue, and the primary ion beam imaging, there's no laser or anything involved. The ion beam is a much harder ionization process. It sputters off those reporter ions as elemental atoms already. So there's nothing we've added in, and because of that, there's not a lot of matrix effects. We've tested basically every tissue in the body through these assays, and I think because most human tissues are made out of carbon, nitrogen, oxygen, and hydrogen, there's very little, chemically, the assay doesn't know the difference of whether we're looking at gut or brain or breast. So generally, there is no matrix effects. But again, it's an important thing to ask when you're doing things like SIMS.
I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. We would also like to once again thank Sean for his presentation and thank our sponsor IonPath for underwriting today's educational webcast. This webcast can be viewed on demand through August of 2019. Leverett will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.